Okay. Hello, everyone, and welcome to this session on practical issues. My name is Ludovico Boratto. I'm from the University of Cagliari, and in this session, we're going to have 6 presentations, mixing, as usual, industry talks and regular papers. So if you are following from home, I invite you to ask your questions as soon as you have them so that we can take the best out of the Q&A time with our speakers. And if you're here, of course, you can use our microphone. So let's start with our first talk, which is an industry talk from Anna Gogleva from AstraZeneca, who will be talking to us about drug discovery as a recommendation problem, challenges and complexities in biological decisions. Anna? The floor is yours.
Thank you. All right. Hello, my name is Anna. I'm a data scientist at AstraZeneca, and I won't talk about vaccines, in case you've been wondering. Instead, I'll talk about drug discovery as a new and exciting field for application of recommendation approaches. So, drug discovery is a very hard problem. It is extremely difficult to find safe and efficient medicines, and it usually takes up to 10 years to discover a new medication. And usually you start with, say, 10,000 compounds, and you end up with one, which is going to be approved by FDA and going to turn into a real drug. And such embarrassingly low success rate is not happening because regulations are tough and we have to jump through many hoops while we're trying to do drug discovery, but mainly because biology is difficult and we don't understand it well enough to be able to manipulate in some sort of predictable and robust way. But anyways, we have to keep going. We need to solve drug discovery and we need to get better at it because there are existing diseases that require treatments, and also there are emerging diseases that require treatment. So the question is, how do we get better at drug discovery? And we believe that recommendation approaches can give drug discovery as a field a massive boost and can help in many places. Because many biological problems are actually recommendation problems in disguise. For example, at the earlier stages of drug discovery, we often want to find causal relationships between genes and diseases. Or we might want to match already existing compounds to alternative disease indications. So this is a so-called drug repositioning problem. And in general, we keep caring a lot about all sorts of relationships between these 3 types of entities: compounds, genes, and diseases. And quite often we want to predict, for example, what's the best gene given a certain disease, or what's the best compound given a particular gene target. Of course, there are a few issues, and in general, when we're operating in the biomedical domain, we are dealing with an extremely large number of entities. There are millions of existing compounds, thousands of human genes. The number varies depending on the year, but still there are a lot. And at least 2,000 human diseases. So it's quite a complex landscape. And biological entities we are dealing with are complex as well. For example, it's not just a single gene that's causing a disease. In many situations, it's a whole ensemble of genes, and they all have complex interactions, and we need to figure out what's going on to be able to develop a treatment. So we need to develop a disease hypothesis. And it might seem reasonable to go and check what's already available in the literature about your problem when you're building a disease hypothesis. But if you're doing it in, sorry, spoilers, if you're doing it in a biomedical domain, you become overwhelmed very quickly because biomedical knowledge is spread across multiple resources. There are papers, there are publications, there are lots and lots of standalone biomedical facts. So some of them overlap, quite a few of them contradict each other. So it's quite hard to build a coherent hypothesis out of this. So what we are trying to do in AstraZeneca, we are organizing all this slightly disorganized and sometimes contradicting biomedical facts into a knowledge graph. It contains both publicly available data and internal company data, such as clinical trials, experimental data. And the graph is quite big. It has more than 11 million nodes and 250 million edges, and it keeps growing as we speak and getting better. Kudos to the team. And our idea was to use graph as a source of context and features for recommendation models. And here I just wanted to show you some early success story, how we are using this in practice with the real and messy, poorly defined biological problem. So here we're interested in lung cancer, and in some patients stop responding to treatment after a while. So this is a phenomenon called drug resistance. We don't know why it's happening, and we need to figure out why. There is a bit of a biology that's known about the situation, but it's not clear, and we kind of have no clue how to treat this. And what we wanted to do is to help scientists find key genes and to do it fast. So a standard protocol in this case involves running a special assay called high-throughput genomic screen. And the main goal of this assay is to reduce the number of potential candidates from a whole genome to just a few thousands of genes. But it is still too many if you want to develop a disease hypothesis and develop a drug. It's just, yeah. Still too hard to target a few thousand genes. So what scientists are doing usually, they go and manually check every single one of these candidates and manually pick the targets that are going to be good ones. This exercise requires a lot of domain expertise, takes a few months to complete, and in general, we just wanted to improve this stage. So what scientists are doing is they're usually looking for an ideal target that fits a bunch of criteria. It should be novel, it should have enough experimental evidence, but it also should be easily validated at the bench and so on. There is a very long list, but yeah, as with many things in life, ideal targets don't exist, and there are no ideal targets in drug discovery as well. But we can look for optimal targets instead, right? So we are trying to model this problem as a multi-objective optimization problem, where we have a bunch of criteria and we are trying to find some sort of an optimal trade-off between them. Some of these criteria are quite simple, others a little bit more vague. And what we are trying to do is to map them to hybrid feature set. And our feature set contains features that we derive directly from the graph using graph structural information. We have metrics such as betweenness, page rank, node degree for each node. And also we compute embeddings that we can use to estimate, for example, similarities between the nodes. And on top of this, we add clinical and preclinical features just to bring in more biological signal to the data set. So, okay, in terms of approaches, we are quite limited because this is a very unique problem and we cannot borrow, for example, from related cancers because every single cancer escapes treatments in their own special way, and we're not learning much from this. So we have to use sort of weakly supervised or semi-supervised approaches. And here we can do still a bunch of things. We can compute directly, for example, non-dominated solutions if the number of objectives is not too big, or we can use stochastic optimization techniques using, for example, evolutionary approaches, or we can go a bit on a tangent and go with implicit matrix factorization approaches using graph connectivity data as a sort of input. So I'll just we'll tell you about the first one because we just have a bit more data to back it up, but we tried all of these approaches and more. So here we're just using this simple implementation of multi-objective optimization. We're using skyline operators to compute non-dominant solutions. And since we don't know, we don't have any training data, there is no way we could learn any weights for our objectives. So we decided to ask our domain experts directly, so what matters to them. And to do this in a nice way, we've built an interactive interface where domain scientists can pick the objectives they like, directions of optimization they like, and run sort of mini experiments and see what's possible given the data constraints. So with this tool, we gave them sort of a framework to think about the problem, but we didn't give them any particular answers. And still, we wanted to validate how well we are doing in general. Are we on the right track or not? Validation is tricky because we don't know what we are looking for. But still, we can do a few things. First, we wanted to estimate what types of features are important for scientists when they are making these sorts of decisions. And here we used limited historical data set, and we tried to model our scientists as a black box classifier and try to figure out which features are the most important to them. So we used two types of feature sets. The first one was mimicking features that scientists typically use when they're solving these sorts of problems. And if you look at this set here on the left, you can find that you can explain predictions just based on one feature quite well. This is not very exciting and a little bit disappointing because we thought the main scientists are doing decisions in a more sophisticated way. But then we brought in the full feature set that we were constructing, including graph-derived, literature-derived, and all other features. And we found out that graph-derived features actually follow the biological ones very closely. And this suggests that in some scenarios when, for example, we don't have expensive experimental data, we can already use relatively cheap graph-derived data and already derive some insights about our problem. Next, we validated our results a bit more formally. So we fixed objectives, generated recommended lists, and put it in front of a group of experts in lung cancer and asked them to annotate our results. And we found out that majority of our predictions were labeled by the experts as novel and credible. We especially like the novel bit in here, means that they were not able to discover these types of solutions before just going with their default strategies. But yeah, we are operating in this biomedical scenario, and ultimately we want biological validation. So we ran a small-scale experimental validation where we took 4 genes from our recommended list and tried to delete it in cell lines that kind of mimic what's going on in lung cancer when it becomes resistant to treatment. And we found out that indeed these 4 genes, they mediate resistance in lung cancer, which is very, very encouraging already. We know there is still quite a lot of things to do, and this is just the first pass of this very simple, very naive recommendation system. And ideally, we would like to close the loop between the experimental validation and the model so that the model learns from biology, if you want, directly. Yeah, so we built this very simple recommendation system, but it turns out to be extremely useful in practical settings. It reduces the number that scientists need to get initial results from months to seconds, and it can be reused for many related problems. And a couple of take-home messages. Yeah, so I was trying to convince you during this few minutes that drug discovery is a very exciting field, and there are many possibilities to apply recommendation solutions there. Of course, it's not trivial, so we cannot just do simple replacement, drop-in replacement. We need to think about our own solutions in this particular space. But with already relatively simple recommendation systems, so we can have a lot of positive impact if you're after positive impact. Yeah, so there are a number of challenges when we're trying to translate traditional recommendation approaches to biomedical domain, and we try to summarize it in our extended deck. So if you're interested, please go and check it out in this GitHub repository. And I just wanted to say thank you to the amazing knowledge graph team in AstraZeneca. And if you're interested in this, please let us know. We are happy to collaborate. Thank you.
Thank you. Thank you, Anna. We have one question online which is coming from Oliver Lloyd, and he's asking, the embeddings should capture structural graph features. What's the benefit of using topological features such as betweenness alongside the embeddings? And he's asking if this could possibly lead to some redundant information being processed.
Yeah, there is a possibility. So I guess we can go from a practical point of view, is just Usually it's much cheaper to compute this overall structural metrics based on the graph rather than trying to compute embeddings on the graph of our size, so it can take a few days to compute. So yeah, this is one thing, but still I would argue that yes, we capture structural information in embeddings, but we capture slightly different aspects of this structural information with additional graph-derived metrics. And another added benefit of, embeddings are great, but it's a little bit hard to explain what embeddings are doing when you're working with biologists, and other types of graph-derived metrics that are a bit more intuitive in this way.
Sure. Okay, let's take one question from the audience.
Hi, thank you for the talk. Really interesting domain because of the inherent problems. What I was wondering is, a big part of the problem seems to be that the experimental results are expensive to obtain, basically, especially if you need to do cell cultures, and high throughput is limited in that sense. I was wondering to what extent you're considering using kind of active learning approaches where you're trying to not just select potential drugs, but also perhaps select drugs for experimentation that will add more information to future trials that may not necessarily be more oriented towards exploration rather than exploitation, basically.
Yeah, that's a great question. That's one of the things that we are trying to do as well. So we want to make recommendations also for potential new experiments. So actually, this drug discovery process, it's very complex, and there are a lot of decisions to be made, and there are many possibilities to build quite niche recommendation systems. And we're probably not going to build a recommendation solution that gives you results from end to end, and we need to have a few of them. For example, have a first pass, recommend the next experiment, like what's the best types of articles I should check. You know, when you start unpacking, you cannot stop.
Thank you.
Okay, we have more questions that are coming online, but I invite Anna to go to the hub, and also you go to the page of the paper on the hub to ask more questions to Anna. So let's thank the speaker again. Thank you.